welcome to the Absolute Recap Biology Edition. I'm your host, Melanie Kingett, and I'll be your guide to scoring a 5. Here at the Absolute Recap, we aim to maximize your understanding and minimize your need for memorization. Each episode will review content, skills, and test-taking tips to help you succeed in May. Your recap starts now. Hi, and welcome to the Absolute Recap Biology Edition. Today's episode will recap the Central Dogma. Let's zoom out. We're in Unit 6, Gene Expression and Regulation, Topic 6.3 and 6.4. Our big idea is information storage and transmission. You've likely heard the expression, you are what you eat. This is partially true. Google the Agouti mouse study on epigenetics if you don't believe me. But the expression would be better phrased as, you are what your proteins produce. This episode will focus on the thematic mechanisms by which genetic information flows from the sequence of DNA nucleotides to RNA nucleotides, and lastly, to a sequence of amino acids in a protein. Let's zoom in. The Central Dogma consists of 2 stages, transcription and translation. I don't know about you, but I could never remember which one happened first. A really simple trick is that the C of transcription comes before L of translation in the alphabet. Now that we've gotten that in order, let's focus on what occurs in each step. DNA is found in the nucleus. It's pretty important, having all the instructions for life and all. The cell cannot risk DNA damage by sending out its only copy of instructions into the cytoplasm. Instead, an abbreviated version of the genetic message is produced. This message is called the mRNA transcript, from the Latin scribere, meaning to write. The M in mRNA stands for messenger. Similarly, colleges request a high school transcript with any student application. This does not represent every grade you've ever received in school, but a shortened and abbreviated version of the most significant grades and courses. We also have transcripts of these episodes. Check out our website for details. And so, the mRNA transcript represents only the portion of DNA which codes for a specific gene. Only one side of the DNA strand serves as the template for transcription at one time. This template strand is also known as the non-coding, minus, or antisense strand. I'm sorry about that. Focus less on its name and more on its directionality. The nitrogenous bases on the template strand are read in the 3' to 5' direction, while the enzyme RNA Polymerase synthesizes the mRNA transcript in the 5' to 3' direction. Remember DNA polymerase and DNA replication? That enzyme also synthesized new strands in the 5' to 3' direction. The mRNA transcript is produced following Chargaff's rules of complementary base pairs, with one exception. Guanine is still complementary to cytosine, but there is no thymine in a molecule of RNA. Adenine will instead pair with a different pyrimidine, called uracil. For example, if the DNA code was 3' to 5', A, T, C, G, A, G, the mRNA transcript would be, pause for you to think, 5' to 3', UAG, CUC. In eukaryotic cells, the mRNA transcript undergoes a series of enzyme-related modifications before it leaves the nucleus through a pore. I consider this the step in which the message is cleaned up, like when you forget to cross your T's and dot your I's, and short handles are added to the physical message. A 3' poly A tail is added, which is just a bunch of adenines. A 5' modified cap is added, which is just made of guanine. And any short, non-coding segments called introns are spliced out. The nucleotides which remain in the transcript are called exons, which will eventually be expressed. And ta-da! We've made an mRNA transcript, a short, single-stranded message of nucleic acids. But you aren't made of nucleic acids. You're made of proteins, and all the products produced by proteins. We need to translate the nucleic acid message into amino acids, a different chemical language. Be careful and watch your words. mRNA doesn't become or turn into protein. Instead, it codes for a sequence of amino acids. Translation of the mRNA to generate a polypeptide chain occurs on ribosomes in both prokaryotic and eukaryotic cells. In short, rRNA, or ribosomal RNA, reads 3 nucleotides of mRNA at a time, called codons. These codons correlate to one specific amino acid, which is brought over by tRNA, or transfer RNA. For example, the mRNA codon UCG encodes for the amino acid serine. This amino acid, like others, is then transferred to the growing polypeptide chain, and the process is continued until a stop codon is reached. The newly synthesized polypeptide is released for further processing and shipping. Some quick math. How many different triplet combinations can you make with only 4 letters? There are 64 different codons. Of these, 1 is a start codon, AUG, which codes for the amino acid methionine, and 3 are stop codons, which do not translate into an amino acid. Since there are only 20 different amino acids, more than one codon correlates to each. Almost all organisms use the same genetic code. This evidence and overlapping codons further supports the common ancestry of all living organisms. Do not memorize any DNA sequences or the amino acid codon chart. Understand how to use it, and practice the process of transcription and translation. To recap, genetic information flows from DNA to RNA to protein. The 3 types of RNA involved in the Central Dogma are mRNA, rRNA, and tRNA. The genetic code is first transcribed into mRNA, which is translated into amino acids. These proteins bend, twist, and fold, and make you who you are. Today's question of the day is about retroviruses. Which enzyme copies the viral RNA genome into DNA for the assembly of new viral progeny? For the answer to today's and future questions, please follow us on Instagram at TheApsoluteRecap. That's the APsolute Recap. If you are a student with questions or a teacher with suggestions, we'd love to hear from you. If you have a biology topic or another AP subject you would like us to cover on the Absolute Recap, please email us at TheApsoluteRecap@gmail.com. That's the APsolute@gmail.com. The Absolute Recap is produced by Brad Kingett with music by Zach Caruso. Please remember to rate, review, and subscribe wherever you get podcasts. Time's up, pencils down. Thank you for listening to the Absolute Recap Biology Edition. AP is a registered trademark of the College Board. Copyright 2020, Absolute Recap, LLC, all rights reserved. Coming up next on the Absolute Recap Biology Edition, Unit 6, Gene Regulation.